So thank you, Kumar, for that introduction, and welcome to this session. So I'd be talking about primary congenital glaucoma and how we have been trying to understand the underlying basis, molecular basis using multi-omics approaches. I don't have any financial disclosures. So primary congenital glaucoma, as we all know, is a developmental disease and with etiology that involves a restricted ocular outflow because of the development. There is a defect in the trabecular meshwork and the anterior chamber angle causing raise of pressures, and the clinical features would have the hapstriae, blepharospasm, photophobia, and so on. We have been interested in this disease just for the prevalence because it's almost 1 in 3,300 in the state in India where I come from, as opposed to other Western countries where it is not a major problem. It almost resembles something in the Saudi Arabians and the Slovakian gypsies. And the reason we have been interested in is because of the population structure, which practices a lot of consanguinity. There is inbreeding, and that raises the prevalence of autosomal recessive diseases. With linkage analysis, few loci were mapped. The first one, of course, was the GLC3A, which harbors the CYP1B1 gene, then the 3D, which is a bit controversial in terms of classical congenital glaucoma because it has other phenotypes, and the 3E later came up with whole exome sequencings. Myocillin and FOXY1 were also mapped. Myocillin, although mapped for JOAG, was shown to be involved in congenital glaucoma. Later, using whole exome sequencings, there are a few other genes. These are not necessarily loci. The loci are not characterized, but these are genes that were found due to sequencings by whole exomes, targeted sequencing, and so on. If you look into the profile of candidate gene across the globe, CYP1B1 obviously has a major involvement because it has been screened extensively as opposed to LTBP2, TEC, myocillin, and FOXY1, which are considered the 5 major candidate genes. Our strategy has been using multi-omics because using the traditional or the conventional approach, we haven't been able to understand or decipher the entire genetic basis of PCG. So we took a multi-omics approach. One was using the genetic basis of using a combination of whole exome, whole genome, and targeted sequencings, followed by understanding the multi-allelic inheritance and interactions, and thereby undertaking a genotype-phenotype correlations. Using the quantitative traits, again, to understand the genotype-phenotype correlations. And the other was to use proteomics approach using the trabecular meshwork, aqueous humor, and then trying to understand novel proteins that are involved, and then linking all of the 3 together. So earlier we had shown, in collaboration with Dr. Hemant Khanna, who is here, that digenic inheritance, that is inheritance of 2 different alleles of 2 different genes, could cause congenital glaucoma, and TEC was one such which was involved in a digenic scenario. And we also found that depending on where the mutation is, whether it is extracellular or intracellular, they have different phenotypes. We found that these variations also have a corresponding physical interactions. As you see that in wild type, they interact together, the TEC and the CYP1B1, whereas with respect to the mutations, when the mutations were created, there is a perturbation of those interactions, which could be resulting or contributing to the phenotype. On the other hand, if we take only the wild type of one gene, the heterozygous component and wild type component of one of the genes, they do not show such interactions. We also showed that since angiopoietin, which is a ligand of TEC, that also has different kinds of behavior in the HUVEC cell lines, and this has already been published. Furthermore, we showed other genes like the serine protease that also interacts with CYP1B1, and knocking down of the serine protease in mice resulted in hypoplastic trabecular meshwork. Similarly with GLIS, which also interacts with other glaucomatous genes like FOXY1, and again, knocking down GLIS results in hypoplastic trabecular meshwork, and so on and so forth. Recently, we also found through our studies on whole exome sequencing that cilia genes, particularly CEP164 and INPP5E, could also be involved in PCG pathogenesis, and they also interact amongst each. There is a physical interaction between CEP and CYP1B1. Taking all of this, we also tried to identify that can we have different measures of genotype-phenotype correlations, and we tried to take quantitative traits not only with respect to the corneal diameter, intraocular pressure, etc., but we looked into the preservation of the retinal ganglion cells and tried to do photopic negative response on these childs. And we found that given the signature of multi-allelic inheritance, there is a difference in the photopic negative response in the children who develop congenital glaucoma as opposed to those who do not. And we also looked into the correlations of those parameters, the clinical parameters with parameters of the PHNR. And we find that individuals or patients who have multi-allelic changes, they have a stronger correlation as opposed to those on your right-hand panel who do not harbor multi-allelic interactions. So in short, candidate genes have 60% unknown. When we use other forms of sequencing, deep sequencing, like using a targeted panel, we reduce that to another 48%. Using whole exome sequencing, we reduce that further, and so on and so forth. But what is interesting is that 20% or so are the co-occurring alleles, as I showed, where they do have corresponding genetic as well as physical interactions. And this is not necessarily the case with congenital glaucoma. Even with very large cohorts of retinal dystrophies, there was a study by Steve Diger and others who showed that even using whole exomes, whole genome, and using copy number changes, they were also having another 30% of the cases which were not accounted for. And so the cases that are not accounted for in our cohorts could also be because there are mitochondrial changes, which we are looking into now. There could be copy number variations, epigenetic changes, which have not yet been characterized. Interestingly, these co-occurring variants, if we look into the whole repertoire of genes, they have multi-allelic inheritance. The red boxes indicate that they are homozygous, blue, they are heterozygous, and yellow are compound heterozygous. So on the z-axis, you have the cases, the y-axis has the genes, and this is just a snapshot to show that all the patients have different forms of multi-allelic interactions. Interestingly, this kind of phenomena is also seen in controls, and controls also have these kind of signatures. And so while determining what actually is the actual signature for the phenotype, we need to kind of eliminate these kind of combinations. Added to that, India presents a lot of ethnic diversity, and that actually increases the problem because many of the data that you find from India are based on the controls which are based on 1,000 genomes or the GenoMat database. But India never participated in those. So we have surrogate populations in those, like for instance, the Punjabis in Lahore, Gujaratis of Houston, Telugus of UK, Sri Lankan Tamils, and Bengalis of Bangladesh. We generated our own controls from a longitudinal cohort, which we have been following up for the last 20 years. And just for the set of glaucoma-associated genes, we found, and all anterior segment genes together, and ciliary genes together, we found that there are 390 or close to 400 pathogenic variants, of which another half of them are present in the controls. Now, what happens is when we overlay this data on the 1,000 genome or others, you find that in the 1,000 genomes, the allelic diversity is much less compared to what you find in the local population. So that's, again, a kind of a red flag that when you consider data from India, you need to ask for the local cohort, and they will have more of allelic diversity, which will render that some of these variants, which are found on the background of 1,000 genomes and nomads, to be false positives. We again, then we looked into the expressions of these genes, and we found that anterior segment genes could be involved in the development of anterior chamber based on their levels of expressions. We also found that these are not present in the adults. These are more expressed in the childhood cases. And when we do these interactions, we find, to cut a long story short, that there are two different pathways that stands out, the hedgehog signaling and the Wnt signaling pathway. And these could be involved in congenital glaucoma. We then took into proteomics, we took cases which are, based on the genetics, we categorized the cases into homozygous, heterozygous, and those without variations in CYP1B1. And we found that there are certain unique proteins which are present in the trabecular meshwork of these patients. And what we found is that homozygotes have an overexpression of TGF-beta, and those without variations, which are PCG, but no variations in CYP1B1, have a higher expression of CYP1B1. These unique proteins, the TGF-beta, are upregulated or enriched in homozygous cases as opposed to those who are heterozygous. And we find a different kind of a pathway because the TGF-beta is regulated by endothelin-1, which is a vasoconstrictor. And what we find is that in cases which do not harbor CYP1B1, we have overexpression of CYP1B1. Those with homozygous mutations usually do not have CYP1B1, and endothelin-1 is overexpressed in the aqueous humor of these patients who harbor CYP1B1 variations, indicating that the pathogenesis could be due to the ECM dysregulation, and which is contributed further by other ECM matrix and the endothelin-1, which actually increases the TGF-beta and reduces CYP1B1. So this is the working hypothesis that we have currently. One, which is through the hedgehog signaling, one through the Wnt, the different interactions of the genes, and there is a cellular death, cell death component also, which is due to the reduction of CYP1B1, overexpression of endothelin-1, leading to TGF-beta. So to conclude, it is a complex disease, which was earlier not thought of as a complex disease, and it has multiple variants with varying magnitudes of effect. So we need to consider these multi-omics approaches, and combinations of sequencing strategies actually revealed signatures, which we have validated some of them, not all of them. And that may reveal ocular angle development and raised IOPs, which are again complex mechanisms, and it could be deciphered through multi-omics approaches. And the functional interactions of genes in the maintenance of the structures may hold key towards understanding of disease pathogenesis. I'd like to acknowledge the patients, the volunteers, the funding agencies, the collaborators. Then thank you.
Thank you, Shubho. We can go for one or two quick questions. If not, yeah.
Just ask a
Yeah,
question.
go ahead.
I was just noticing the great work you've done with proteomics, like from trabecular measurement. It's impressive. I'm not aware of many people who've done that. Do you see any correlation between the protein levels that you measure in the eye with sort of like systemic protein measurements? Like how much you actually measure systemically? Do you see traces of those signals like peripherally?
No, what we saw is with regards to, the proteins, what we had extracted, we had loaded almost equal amount, both in cases who harbored mutations in CYP1B1, because that was the categorization, and those who did not have. So these did not have were the ones who would be considered as normals, right? So the proteins were, the protein amount was the same. Signatures were different, because if you see in that pie chart, I rushed through it, there were certain unique signatures which were specific to those cases who harbored mutations, those who did not harbor. Yeah, but there were majority of the, I think 2084 proteins were common to them, but the number which will be unique would be 100 or so among the homozygous cases, and those without mutation would be around 40 or so.
So I was thinking about the potential to actually use those biomarkers.
Right. Yeah.
In the diagram where you showed that you were looking at genes other than CYP1B1 as well, and you showed that there are patients in which there's CYP1B1, and along with that there'll be some anterior segment gene like
Correct.
a PITX. Were their phenotypes distinct in any manner, or apparently they were the same?
No, these were cases who were classical cases of PCG.
Okay.
So during our inclusion itself, Those who had a variable phenotype were not considered. So within the classical cases of phenotype, we wanted to see what is the extent of the genetic diversity that we are able to see.
And were those variants benign or SNPs, or you only marked them because they had some sort of a higher pathogenicity?
These
or
are all the rare alleles which are pathogenic with a Revel score of 0.5 and above. Yeah, it was there somewhere in the bottom. Yeah.
Thank you.
Thank you, Dr.
Sure. Thank you very much.